Hello, everyone, and a big welcome to all our attendees. Thank you so much for joining us. Criterion Edge is very excited to present our webinar today. The topic, a practical guide conducting systematic literature reviews in support of IVDR readiness. Our first presenter today is Criterion Edge's president, Lori Mitchell. Lori is founder and president of Criterion Edge, a global medical and regulatory writing and safety services firm serving the medical device, pharmaceutical, and biotech industries. Lori has over 20 years of experience in medical writing, safety and pharmacovigilance management, and regulatory reporting. Having provided regulatory and medical writing solutions to many pharma and medical device companies, both large and small, she is a proven leader in designing practical strategies to meet current global regulatory challenges. Lori is a published author and holds a Master of Nursing from UCLA. Our second presenter is Dr. Sarah Chavez, Principal Medical Writer with Criterion Edge. Sarah has a bachelor's in biochemistry and a PhD in molecular and cellular biology with 25 years of laboratory bench experience and has taught at the university level for the past 15 years. She has extensive scientific writing experience in an academic environment as well as for several large textbook publishers. Sarah's strong background in basic research and her extensive experience with regulatory writing will allow her to assist companies with IVDR readiness as part of the Criterion Edge team. Now, before we begin, I want to mention a few housekeeping items. If you experience any technical difficulties in the duration of this webinar, please use the chat option on your Zoom menu bar and send us a message, and someone will try to do our best to resolve it. If you have any questions about our content today, you don't have to wait until the end. You can type in your questions at any point in time using the Q&A option also on your Zoom menu bar. And we'll run through as many of them as we can. If we don't get to your question, we'll try to get in touch with you afterwards. Lastly, this webinar is being recorded and will be available to view on our website afterwards. And you will also receive an email with the link to the recording as well as the slides. Before we dive in, I wanted to share some interesting results from the responses we received when you guys registered. Nearly half of you identify yourselves as a part of the regulatory affairs and quality area. And I wanted to say, no matter what your background is, I'm sure that you will find this presentation very useful. And now let's take a quick moment to take a poll that's going to show up on your screen right now. And we'd like for you to answer this question for us. Currently, how aligned are your internal processes and documents, quality system, tech file, risk, PMS management, performance evaluation process, etc., with IVDR requirements? And I'll just give you guys a few seconds to finish responding. Okay, well, thank you everybody for your responses. It looks like most of you feel like you still have gaps in your processes. Well, I certainly hope you find that this information presented in the webinar will give you more confidence and give you more tools to be best prepared. And with that, I will turn it over to you, Lori.
Thank you, Jessica, and welcome everyone to this presentation on how systematic literature review can help support IVDR readiness within your organization. We hope this webinar will present you with practical guidance on how to conduct a methodologically sound systematic literature review and insights into how published data is leveraged for critical IVDR required reports, such as the Scientific Validity Report, or SVR. First, I'll say, though, that I'm sure that many of us are attending this webinar from the comfort of our home offices, and we thank you for taking the time to attend. And I'd like to give a special shout out to our small two-legged and four-legged office assistants who may also be attending. Welcome. Okay, let's get started. Companies need data to inform organizational strategies and direction, to guide critical decision-making, and in this case, to meet global regulatory requirements. but performing systematic literature reviews can present multiple challenges to a team. For example, is there access to the right expertise, such as a medical librarian, to consult with and design a sound search strategy? Are regulatory compliant processes in place that support robust literature search, screening, and selection activities? And lastly, even with all of the above, executing the steps of a systematic literature review requires people. Even with tools and technology, there's no way to remove trained human eyes and brains from the process. The screening, review, and selection steps take time and people, both of which may be in short supply within a company. Bottom line is that maintaining a consistent process while increasing efficiency is difficult when there's a lack of available resources, or the steps of the process are ill-defined, or manual processes are used that are slow and prone to error. In today's regulatory landscape, adopting a transparent, reproducible process for literature reviews is essential. The objective of our session today is to review currently accepted systematic literature review practices and provide guidance for designing and conducting a rigorous literature review from search strategies through screening, data extraction, and reporting. At the conclusion of this webinar, you will know how leveraging published data can help fill knowledge gaps, meet IVDR expectations, and inform internal decision-making. Why systematic literature review is superior to a simple internet search, steps for conducting a methodologically sound literature review, and by examining a relevant case study scenario, how data obtained through systematic literature review can be used to support IVDR required reports. We'll start with a description of the framework of a literature review and also examine how conducting an internet search is much different from the systematic literature review process. Next slide, Jessica, thank you. A systematic literature review is a clear and methodologically sound plan for the identification, retrieval, selection, appraisal, and weighting of published data. Systematic review methodology, I should say, is nothing new. It has long been the gold standard process for evidence-based research, especially in the academic and professional arenas. The data derived from this method have long been used, for example, to develop clinical practice guidelines and recommendations, and describe and support academic-based research initiatives, among other applications. Those same rigorous principles are now being mandated by global regulatory authorities and have been adopted by pharmaceutical, medical device, and IVD manufacturers as best practice. But first, let's establish the key differences between an internet search and a systematic literature review, because they are certainly not equivalent to each other. Internet searches should be considered ad hoc because they don't follow a predetermined plan methodology as a systematic literature search does. This graphic presents the high-level differences between these two types of searches. Let's ask ourselves these questions. First, can the search results be considered comprehensive and thorough, extensively scoped across all relevant literature sources? The answer for an internet search is definitely no. The results are dependent upon the limited capability of internet search technology, the availability of the published literature, and the relative skill of the searcher. In contrast, a comprehensive systematic literature search employs a search strategy designed by a person knowledgeable in advanced search terminology and is run on a robust software platform that supports advanced search capabilities. Next, can multiple literature databases be searched? No, not on the internet. Google Scholar is not really considered a database, and PubMed is the only major database that can be accessed on the internet without a paid subscription. However, again, it is assumed that a systematic search can be run on a platform with access to all relevant databases, minimally Embase and Medline. Can the search platforms support flexible search terminology? No, the internet cannot. Searches are dependent on keyword selection, or in the case of PubMed, only the and or search functions are supported. To achieve comprehensive and focused results from your literature search, the search strategy must use advanced mesh terminology and search syntax. This requires the use of a robust search platform, as I already described, such as ProQuest Dialog. Is the methodology clearly documented and reproducible, reliably yielding the same or similar results each time? That's a definite no for an internet search. However, a well-documented search strategy conducted on a search platform will reproduce similar results again and again. Can objectivity in screening and selection of the literature be demonstrated? This characteristic relates not to the literature search, but to the criteria used in the literature screening and selection process. It is possible to apply the same rigor and level of documentation to the screening and selection of literature identified via an internet search. However, if the search process itself is already questionable, it hardly matters if the screening and selection process is well described. The battle for objectivity is already lost, so to speak. Now let's dive into the details on the methodology of the systematic literature review. Here's a bird's eye view of the basic steps of every systematic literature review. Develop and define the research question or questions that are under investigation. Design the search strategy and conduct the search using all relevant databases. Screen all literature references returned from the search by applying predetermined exclusion and screening criteria to define the selection process. Extract relevant data from those included full-text articles that were selected during screening. And finally, synthesize, organize, and analyze the extracted data into a report to answer the research questions. So let's get started with step one, which is the scoping your search and the screening process. The initial task of the scoping process is to define the research question, which is really a critical first step in the overall scoping of the literature review process. You should identify the purpose of the search. Basically, what do you need to know? Then establish clear research questions that will support that purpose. Identify where to search by selecting relevant databases such as Embase, Medline, SciSearch, and others. Determine if other sources of data will be considered, such as unpublished literature or web content. A well-defined scoping process provides the foundation for the entire literature review. You will know that this scoping step is complete when the search questions are clear, the literature search planning process has been thoroughly documented, and the search strategy is well defined. Key takeaways for the scoping process are: take the time to determine clear research questions and document your process for transparency and reproducibility. Consider developing a literature search scoping worksheet to guide and document the process. At Criterion Edge, we have developed such a tool to support our client collaboration and the subsequent kickoff meeting of every major writing project we are engaged with, most commonly the clinical evaluation report. We found it to be a very useful tool, and our clients like it, and certainly we do too. Step two, the search. Using the framework derived from the scoping exercise, the next step is to basically design and run your literature search. Here you'll need to identify key search parameters, such as relevant search terms, for example, product names, PICO terms. And if you're not familiar with the PICO acronym, it stands for patient or population, indications or interventions, comparison or comparator, and outcomes. Using focused and relevant PICO search terms to support the scientific validity report is especially important. Determine exclusion criteria to include in your search terms, such as foreign language or animal studies, technical articles, etc. These are common types of articles you can exclude from your search results by using the right search terms and proper mesh syntax. You'll need to establish certain search limits, such as the time range, study type, or levels of evidence. For example, you can exclude abstracts or conference proceedings. A couple of key takeaways at this step are: use a librarian with experience designing medical information search strategies, maintain a record of all literature results returned by the search. This documents that all potentially relevant articles were considered for inclusion. Step 3, screening. Here I highlight a few best practices for the literature screening process. Let's start by talking about the use of robust methods to identify and select the appropriate literature to answer your research questions, to minimize selection bias, and introduce some time-saving efficiencies to this very labor-intensive part of the process. First, successful screening depends heavily on clearly defined eligibility criteria, which you identified during that scoping step. Literature screening is a very human part of the overall process, and screeners should be trained on and strictly follow those pre-established eligibility criteria, because those criteria translate into screening decisions by the humans doing the screening. Strict adherence to pre-established screening criteria, along with thorough documentation of screening decisions, provides evidence that selection bias was minimized. Transparency and reproducibility are critical components of the screening process, and you should definitely document your screening decisions to comply with global regulatory standards, such as those outlined in MedDev 2.7/1 Rev 4. Lists of excluded references and the reason for exclusion are common and expected reporting requirements. Now, I'm sure it won't surprise anyone to imagine that the volume of literature to be screened can be huge. Sarah will address this issue in her remarks, but I will say here that one strategy to manage this challenge and make it more efficient is to use a multi-stage screening process where the first level of screening includes review of the title and abstract only to identify those more straightforward screening decisions, such as, is the article in a foreign language, is it technical data only, or is it not related to this research question or device? This first level of screening saves time by identifying the obvious includes and culling out those obvious excludes. Those references left fall into that maybe pile, where a review of the full text article is necessary to determine the final screening decision. Taking this approach can save you time and money and expends less manpower during the screening process. Data extraction should be also prospectively planned and consistent. As I mentioned earlier, for the scientific validity report, you would extract PICO data that serve to inform and elucidate the questions for your review. Remember, patient, indication, comparator, outcome. Another critical element of data extraction is the identification of relevant clinical safety and performance outcome data, as you would extract these data for use in a clinical performance report, for example. It is at this step that you would appraise the extracted data sets, from each article using an established appraisal methodology, such as one developed by the Oxford Center for Evidence-Based Medicine, OCEBM, or the method we commonly use at Criterion Edge, the current Global Harmonization Task Force guidance document, which is a very robust appraisal tool. We like it very much. A little tip here, the use of predefined, easy-to-use data extraction forms is advised, as it helps avoid human error, which often arises out of manual processes. And certainly data extraction out of articles is a very manual process. Using data collection forms can ultimately result in cleaner data and greater efficiency. These extracted data can then be used to populate relevant tables for analysis in your report. Finally, step 5 of the systematic literature review process addresses the documentation and reporting of your screening results. The PRISMA reporting structure is a clear, systematic flow diagram that documents the results from each step of the literature search, screen, and review process. Documentation requirements may vary depending on where your report will be submitted, but typically will include some or all of the common requirements on the PRISMA shown on this slide. A PRISMA flowchart is typically included as part of your documentation to regulatory authorities and major journals. If you use a screening software platform such as Distiller SR, which we do, creating a PRISMA flowchart is automatic and easy. The key takeaway is this: as a rule of thumb, your documentation should be sufficiently detailed to support full replication, so the importance of transparent reporting cannot be overstated. Well, this concludes my part of the presentation. Thank you very much, and I hope you found the information useful and relevant. Before Sarah discusses these concepts further in a great case study scenario, I'll hand it back over to Jessica for a poll question. Jessica?
Thank you, Lori. Let's take a short break to just take another quick survey. You'll notice the question pop up on your screens now. And our question is, what are the biggest challenges faced by you or your company for IVDR readiness? And you can choose more than one. So I'll just give you all a few seconds to answer the poll question for us. Just a couple more seconds as more responses are coming in. Okay, thanks everybody. It looks like a majority of you are experiencing people resources issues where you don't have enough bandwidth and possibly experiencing knowledge gaps. Definitely something that we've seen quite commonly. So thank you for all your responses there. At this point, I will turn it over to our second presenter, Dr. Sarah Chavez, who will give us some insight with a case study.
Thank you, Jessica, and thank you, Lori, for providing such a great overview of the systematic literature review process. IVDR includes significant global changes, specifically new language regarding systematic literature review. So while the overall focus of today's webinar is how systematic literature review can support alignment with IVDR regulations, it's also important to point out that MDR was published in parallel with IVDR. So having prior knowledge and experience with MDR guidelines will be extremely helpful for a successful transition to IVDR. So now let's turn our attention to a case study to help illustrate how systematic literature review can provide the critical background and really the foundation for building the supporting documents required by IVDR. The case study will focus on the role of systematic literature review in writing a scientific validity report, or SVR, But the same general principles can be applied in preparing your analytical performance reports or the clinical performance report. So in this scenario, a company is preparing to write a PER for their HIV diagnostic test, which is a Class D IVD designed to detect HIV in blood samples using ELISA. They recognize that writing a PER will require systematic literature review. So their gap analysis began by taking a closer look at their existing supporting documents or feeder documents. And in doing so, they realized that the existing draft of their Scientific Validity Report, or their SVR, did not meet the requirements for IVDR. In fact, they were missing a complete systematic literature review. So this is an important point to keep in mind as you're getting started. A thorough review of your internal documents is a crucial first step for assessing your own IVDR readiness. If your SVR does not include data derived from a robust and a well-defined systematic literature review, your PER will be missing that all-important foundation that you need to establish the scientific validity in an objective and a reproducible process. So our strategy would be first and foremost to design a targeted systematic literature review to establish the scientific validity of using ELISA-based HIV testing. And that will bring the SVR into alignment with the updated reporting requirements. So as you're evaluating your own IVDR readiness, keep this in mind. In this scenario, the company requested our assistance in writing a PER. However, if their supporting documents are not in alignment with new IVDR guidelines, their approach is flawed before they even began. So they must be brought into alignment before the process of writing a PER can even begin. As Lori discussed, the first step of a systematic literature review is defining the question. We need to identify studies that establish whether ELISA is a scientifically valid approach for the detection of HIV in blood. And that requires establishing that ELISA-based testing is considered state-of-the-art. So what do we mean by that? IVDR mentions the term state-of-the-art or SOTA at least 20 times, but it never actually defines what SOTA means. So this is where we can turn to MedDev Rev 4 for some guidance. In MDR, the SOTA is broadly defined as the current state of all competitive treatment options. So if we apply this basic concept to IVDR, you can think of SOTA as describing the current state of all competitive diagnostic options. So in that case, our comparators would include other ELISA-based diagnostic assays, including our subject IVD, as well as other competitive tests. So through our systematic literature review, our goal is ultimately to identify high-quality published research studies that use ELISA to detect HIV. The data from the SVR will be incorporated into the PER, so we want to ensure that our research question generates a robust and an unbiased data set to support scientific validity of the test. So in effect, our research question can be very simply stated as, is ELISA a scientifically valid diagnostic methodology? So then our next step is to think about how can we set up a successful search strategy to answer this question. And a truly effective systematic literature review must be very purposeful in the selection of the search criteria. The search criteria you select here will be crucial for retrieving the most relevant publications in a reproducible, unbiased, and a well-documented approach. So the search must include well-defined limits to ensure that the search is targeted and the results are manageable when it comes time to screen these articles for inclusion. Some of the things to consider: what are the key PICO search terms that will help you identify and locate the most potentially relevant studies? But also, what limits do we want to place on the time frame? For something like HIV diagnostic testing, the literature may date back several decades, whereas with something like COVID testing, you would expect to have a much more limited search range. That being said, more is not always better. A longer history in the published literature or a large number of comparator ELISA-based HIV tests on the market means that your search may result in quite a few articles that must be screened by eye in the next step. And that screening can be extremely time intensive and must be well documented as part of the new IVDR requirements. So this step is really where you want to be certain that you've chosen the best possible search limits and filters to generate a targeted yet robust pool of studies. But let's not forget the importance of where you're searching. So in this table here, in Lori's, this is going to give a side-by-side of PubMed searching versus searching in an advanced search platform. In Lori's presentation, she explained that internet and PubMed searches are not the same as an advanced search platform such as ProQuest Dialog. And as you can see here, a robust search platform offers several distinct advantages. If you take a look first at the table on your left, these are the results of a PubMed search. You can see that the search terms used resulted in a large number of records retrieved. If you simply search for HIV test diagnostics, you get well over 6,000 hits. However, changing the order of the words to put diagnostics before test only retrieves 737. So clearly the syntax of the PubMed search strings is not particularly precise if we see such a dramatic difference. Comparing between search 4 and search 5, which are highlighted here in that table on the left, the only difference is whether or not you use the term diagnostic as a singular or a plural. The results literally double if you drop the S in the term diagnostics. So even if you use the most obvious search terms of ELISA and HIV, you're still left with well over 1,200 articles to screen. So now let's take a look at the table on the right. Even when you compare the numbers to what we retrieved with an advanced search platform that covers both NBase and Medline, these results automatically appear much more manageable. In this case, you can cut the number of records down from 1,200 down to 634, and by further refining your search strings, you can get the final number of records down to a much more manageable 58. And remember, you're responsible for screening all of these articles, and you must document your reasons for exclusion. Clearly, screening 58 articles is preferable to screening over 1,200, but also consider how many of those 1,200 articles were completely irrelevant or off target. Screening is arguably the most time-consuming step in this process, so a little extra clarity in your search strategy up front will save you a ton of time and also ensures that the articles are much more targeted and relevant. So now let's take a look at what the screening of these articles actually would entail. The process of screening must be well documented and reproducible, as Lori mentioned, so organization is crucial. Reasons for exclusion must be documented for each record based on your predefined exclusion criteria. You may have some simple discrete exclusion criteria, such as full text availability or language or sample size. These can be documented as part of the screening log to allow you to keep track of the exact reason for excluding each of these records. Other exclusion criteria may require significantly more time or effort to assess. Record 9, for instance, it appears to meet the initial search screening criteria you selected. It was published within the past 5 years, English full text is available, and it's a randomized control trial conducted on 500 patients in the EU. However, a review of the full text article would tell you that the study failed to define the type of HIV screening test being used. Because the SVR is focused only on ELISA-based testing, we would exclude articles that include mixed cohorts or alternative technologies such as PCR-based testing. So this scenario can be relatively common, particularly if the focus of the article was on HIV itself and not on the testing technology being used. There may still be extractable data regarding the accuracy or the sensitivity or specificity of ELISA HIV tests contained within these articles. So the issue, though, is that it could be difficult to determine without full text screening. The benefit of a more targeted search is that you should have much fewer off-target hits to exclude in the level 1 screening. So that will allow you to spend your time reviewing full text of these articles with potentially relevant extractable data. So now let's take a look at what we mean by data extraction. Extractable data is ultimately the goal of systematic literature review. You want to identify relevant, high-quality studies with extractable data that allows you to establish scientific validity of ELISA-based HIV testing. For the SVR, this could include information about the target of the different ELISA-based assays. Do they all use the same antigen or the same antibody combinations? Different ELISAs may use different targets, so that's something to consider when you look at competitor assays compared to your own test. This may also explain potential false positives, so that would be the type of data you would want to extract to compare between the studies. If you were instead preparing a clinical performance report, this may include data such as sensitivity or specificity. How many false positives are observed for each test? If you have a 99% specificity, that tells you that 1% of the positive results are potentially false positives, and that could be a function of the antibody or the target selected. What are the levels of viral titers that are detectable using ELISA? You may be able to address this question by reporting the sensitivity of the assay to get a sense of the number of false negatives reported in clinical settings. This may be an issue with the viral titers, with the test modality itself, or with some other reasoning. So it's important to record other details about the assay in order to make direct comparisons. If you were preparing an analytical performance report, you will want to extract data specific to the test itself. Does it require a certain volume of sample? The volumes may have an effect on the overall sensitivity of the assay, so this should also be noted if you plan to compare between studies and assays. So after extracting these data from each study, you must now be ready to document the overall strategy and the process used itself. So the final step of a systematic literature review is to report your process, to document how you've arrived at your final data set. This PRISMA flowchart must present a strategy that's methodologically robust, unbiased, and reproducible. The goal is to show that you've identified and screened articles objectively because these are the data you'll use to inform your final conclusions. So the systematic literature review ensures that you can be confident in the studies you've identified and the criteria you've selected. And you can use this data to establish scientific validity of ELISA-based HIV diagnostics. This is really the purpose of a systematic literature review. It provides the data necessary to develop IVDR-compliant SVR, but it also ensures that the conclusions you make have been derived from a reproducible and a well-documented process. The end result is that the company has a systematic literature review to support the development of an IVDR-compliant SVR. They can now use this SVR to support the development of their PER and be assured that their PER is now in alignment with the new IVDR guidelines. So I hope this case study has helped us with sort of illustrating how systematic literature review can be used to help your company with developing more IVDR compliant technical documents as a first step towards developing your PER.
Thank you, Sarah. Before we conclude, I just wanted to remind you guys to start sending us your questions. We'll be doing our Q&A very quickly, very soon after. We hope at this point you have an understanding of the systematic literature review process and why it is a required standard for all notified bodies and other global regulatory authorities. We hope that you also understand how to conduct a systematic literature review from scoping through data extraction and reporting, how to implement those required steps of the systematic literature review methodology within your own organization to meet IVD regulations, and how the data identified from a review of the published literature can support your organization's IVDR readiness. If not, please start sending us your questions. If you have any follow-up questions, you can always reach out to Lori or Sarah. Our contact information will show up on the conclusion slide, so stick with us. If you want to know more about our services and want to schedule some time to talk to Lori, please let us know and we'll gladly set that up for you as well. At this point, I'm gonna set up a screen to start populating our questions for Lori and Sarah. All right, and Lori, Sarah, can you both see the screen?
Yes.
Okay, our first question here is, could you give another example of the differences an approach to a systematic literature review for an SVR compared to an analytical performance report or clinical performance report?
Okay, so let's take, for example, COVID and the new COVID tests that are being pushed onto the market right now. A scientific validity report would address the association of a particular analyte with the clinical condition. So this is really where you want to include the results of clinical performance studies, consensus or expert opinions, and the guidelines that really let you say for certain that the analyte you're using is a scientifically valid approach. So this would be where you would show that SARS-CoV-2 infection is clinically associated with COVID-19, the COVID syndrome. So you could also use data regarding symptomatic versus asymptomatic carriers of the virus. Whereas an analytic performance report, that would ensure that the analyte could also be reliably measured using the methodology you've chosen. So that could include data regarding RT-PCR for the nucleocapsid gene, which is how we're typically, the method we're using to reliably detect coronaviruses in many cases. So information about false positives or false negatives. The clinical performance would refer to the ability of the test to yield results that are correlated with the clinical condition in accordance with a target population or a specific intended user. So you could compare your PCR-based test for COVID with other approaches, like using the antibody or the antigen-based testing, or you may compare nasal swab approach versus the newer saliva-based testing, something to that effect.
Okay, great. Thanks, Sarah. Our next question, if you guys can see on the screen, is for well-established analytes such as hemoglobin or glucose, is an SLR really needed to establish the scientific validity? These types of well-known analytes and their scientific validity are already described in international guidelines and recommendations.
Sarah, I can take that because I've got a couple of ideas on how to answer that, if that's all right.
Oh, sure.
Okay. The short answer is we hear this question a lot with regards, do we really need to do this because of these are well-described analytes, well-described devices, such as in the medical device world. And the short answer is to do scientific validity, state-of-the-art. MDR and IVDR both require a systematic literature review to look for that information. Now, let me just be clear about that. In the state-of-the-art section, there are really two parts to that. One part of the state-of-the-art is to describe the state-of-the-art of the technology that you're discussing, your analyte, the technology of your device, and its evolution to its current state, where it began, the patient population that it's intended to service, and so forth. In that part of the state-of-the-art section, it's very common for the writer to run a search to look for guidelines and to look for that freely available website contact on professional guidelines and academic guidelines to describe that background of this systematic literature review, of the state-of-the-art of your device. That is not, does not have to be per IVDR and MDR guidelines. That part of the search does not have to be methodologically robust because it's essentially the writer doing research on their own to describe this background information. However, the other part of the state-of-the-art section, which does require a methodologically robust approach, is to look for data, competitor data, or pooled data. Your device may be in a pooled data set, but it can't be determined, so pooled data is all right. But when you're looking for data to set your safety and performance acceptance criteria for your outcomes, that is establishing, if you will, the bar for your, in which all of your devices data will be compared. That's done in the scientific validity section, where safety and performance acceptance criteria are determined for your safety and performance endpoints for from competitive data. Because that's a data set, that does require a methodologically robust process, because now you're handling data and you must look for it without bias, describe it, weight it, and appraise it. So I hope that helps.
Thanks, Lori. Our next question is, what is the relevant sound number of publications selected for review?
I can take that as well, Sarah. Let's be also clear about when you're looking for, when you run a search on your device and that search comes back with 300 articles. IVDR and MDR require that you look at all 300 of those articles. You don't get to pick a number. So you have no idea. This is the little bit of the devil is in the details, is that when you're using a robust platform, oftentimes your librarian or someone, you can do scoping searches to see what kind of numbers you might be getting back. But at the end of the day, when you push the button and you do a search, that search syntax and search strategy and protocol must be published in your PER. And if it says that 252 articles came back, human eyes must look at all 252 of those citations and references and document inclusion, or if excluded, the reason for exclusion of every article.
Thanks, Lori. Our next question up on the screen. My commercial team tends to have a very broad scope. What recommendations do you have to help them narrow their review questions?
Again, this is Lori. I'll be a little smart alecky here and just say one recommendation would be is to don't invite your commercial team to your scoping meetings. That would be that, and that's because they're not clinical in nature. They may not be clinical in nature. Their focus is something else. If their focus is on a broad scope from a marketing perspective and so forth, I get that. That's their job. But for the purposes of putting together a PER and the supporting reports that feed into a PER, such as the SVR and the CPR and the analytic report, you have to narrow your search in such a way that the research questions are very focused on the task at hand. If it is too broad, it will be, if the focus of your research questions is too broad to try to catch things, you'll catch a lot of junk, essentially, that you'll have to eventually screen out. So that, I would say that the recommendations is to stay focused on the task at hand, which is in a scientific validity report, looking for competitor data and background information on your device.
Thanks, Lori. The next question, if a small company with no librarian, I guess they're talking about being a small company with not having a librarian or access to a librarian, are there sources available that can support search development?
Sarah, I don't know if you've got any ideas, but I know that we certainly do hear from our clients and colleagues out there that they will try to, if they have academic connections, they'll often go use those academic connections to try to access more databases. Because I would say that, while PubMed is a robust database platform, it's on the internet, you're certainly limited to search PubMed with a very limited mesh terminology. Mm-hmm. And as such, you're going to get a lot of false hits. I'm not saying that you must use a robust platform and you must use a librarian, and you must search multiple databases other than PubMed, which is essentially, by and large, it's essentially Medline. Those are acceptable. You just must be prepared for the results, because the rule in IVDR, it's very specific. Whatever your search results are, if it's 50 or 500, IVDR is very plain. You must review them all and screen them all.
And as someone who's worked in a lot of translational medicine style research projects, I know that we consult fairly regularly with our med school librarians. They're able, when we're preparing either patents or some of our clinical reports for getting our clinical trials approved, or even writing it up, we routinely consult with our med school librarians to help us better design our search strategies to make sure that we're getting sort of that same type of robust data that you wouldn't need for IVDR.
Okay, thanks, Lori and Sarah, both. Our next question up here, it is, can I base my SOTA on existing free reviews that I find only on PubMed, or should I find a way to get my hands on articles that are not available for free? Since it is state of the art, can I just review material from the past 2 years?
So this goes back to the question that I answered a couple of questions ago, which is there's really kind of two parts to the state of the art, and one is providing that background information. The maturity of technology, the patient population, the clinical conditions in which the device is relevant to. All of that information can be searched for by the writer themselves, and doesn't necessarily need to be part of a formal search for that kind of background information. Now, that being said, and then if you get the articles for free, or you certainly look at professional guidelines and so forth, that's certainly an acceptable strategy. Now, the second question here is, since it's state of the art, can I just review material for the past 2 years? I would say that I don't think that you should be so confining. What would be the standard is to look for the most relevant and up-to-date guidelines, and 2 years may not be enough. I think you need to be open-minded about searching for those articles that are great sources of information to provide you with your background information on the state of the art.
The other issue you might have is that if you've conducted your entire literature search and now you're trying to come up with exclusion criteria, not having full text available because you didn't buy it might not be a justifiable reason for excluding an article, particularly if it reports on particular safety concerns or alternative treatments or something along those lines.
All right, thank you guys. Next question, which rationale do you use to exclude foreign language as databases embed national journals?
So if the question is, what is the rationale for excluding foreign language articles that we would put, that is a fairly standard exclusion criteria, certainly in the MDR world. And I would imagine it would be just as acceptable in IVDR searches as well. It has been established over the last few years that notified bodies are not requiring companies to pursue translation of articles that are not available in the English language. So if the article only looks like it's available in a non-English language, and you think you can get along without it, in other words, because you can't really even tell what it's about, it's okay to exclude them, with the exclusion criteria being foreign language.
We have had some clients who we've worked with who are based in different European countries though, and in some cases, if it was a very pertinent article and they were able to read it, they were able to have someone on their team help us with the particular data extraction. So you don't necessarily have to exclude it if you know, if you have someone on your team who is able to translate it for you.
Right, thank you. Next question, and I want to just make a quick reminder, you can continue to send us your questions. We do have another 5 minutes left on this webinar, and if we don't get to them, we'll certainly try to reach out to you afterwards. Our next question is, do you think a literature review should be supported by a written plan?
So by literature review, we're talking about the whole methodological process. That's a simple answer, yes. It must be supported by a written plan. Now the plan doesn't, let me just be clear though, the plan doesn't mean like a formal document that you rev within your department. It should, however, be described in your PEP, at least addressed in the PEP. It's often because the PEP is a plan. And you can have a subsection in there to discuss the methodology of the type of methodology you'll use in the support of writing your PER. So it doesn't need to be formalized as a standalone report or plan on its own, but the review process should be clearly described in writing within the report itself, and is usually backed up by a plan that's part of the overall performance evaluation plan, like a subsection of it.
Okay, thanks, Lori. Next question. One big takeaway from the MDR is the need to include information from relevant guidelines. Are there any tips or such helpful resources to easily find relevant IVD guidelines?
Sarah, I'll let you weigh in on this one, but I would also say that, that it's, use your background and experience to know those professional sites, but go ahead, Sarah.
Yeah, that's the main point I think that's important to remember, is that in my experience, Embase and Medline and even the most targeted searches may not retrieve the most updated guidelines or position statements from all of the relevant clinical organizations. So I tend to use supplement with additional target searches in many cases of those particular society websites. So like for HIV testing, you would look to see if there's any particular guidelines from the European Society for Disease Prevention and Control, or CDC guidelines, or something along those lines. You'd want to make sure that you sort of identify larger professional societies and just be ready to go directly to their website.
So then just to finish, this is again to emphasize that when you're writing the state of the art, ad hoc searches to get this kind of background information are very acceptable and a common part of this process. It's again, the methodology must be very robust and transparent when you're doing searches in which you are gathering data.
Thanks, Lori. I think we have time maybe for just one more question, and I know we have a couple that went unanswered, so we'll definitely reach back out later. So the next question is, for a very novel technology, what tips do you have to perform relevant searches?
Sarah, that's you.
Yes, we've encountered this quite a bit as part of the team that's developing some saliva-based COVID testing. You pretty much have to look at what is the most relevant comparator. You may not have a technology that's a direct comparator. Like for instance, I was working on a technique called RNA-scope, where it's using RNA probes as sort of an alternative to in situ hybridization. It's the only technology of its kind, and it's a very novel technology. So if I were to be doing background searches for that, I would probably have to use in situ as sort of my comparator, just to give sort of a sense of, in a clinical setting, what's the detection range for a particular protein using an antibody, and then, or you could also use qPCR and look at the how other ways of quantifying RNA. So it sort of depends on your research questions, but you do have to cast a sort of broader net, so to speak, if you don't have a direct comparator that you're dealing with.
Thanks, Sarah. We are out of time today, so I want to thank you both, Lori and Sarah. But before we close out, I just want to take another quick poll and see how you guys are feeling about this webinar and just generally how confident are you feeling now that you've been through this. Please just take a couple seconds before you jump off to answer this one question for us. How confident are you feeling in your internal performance evaluation processes and IVDR readiness, with 1 being not at all confident to 10 being extremely confident? And while you answer that, if you have any questions about our content, any more questions that you think of, or you would like to learn about our services, we do have a free consult available for all our attendees. The contact information should be showing up on your screen. It's consult@criterionedge.com. Thank you, everybody, for attending. This webinar is recorded, and it will be available very soon after we close out here on our website, and you will receive an email as well with the recording and also the slides. I want to thank you all for joining us again, and thank you, Lori and Sarah, again. You guys have a wonderful day, and stay safe.